drink is retained, but then you don't have any problems the next day. So that philosophy, we said, okay, when we started looking around at all the current advisories, whatever were there, we found 90% of them are all home remedies. You drink more alcohol, you have, and then you have drink coffee, you sit in the sunlight, you eat curds, all sorts of stories. And then in US, there are almost about 200 OTC, over-the-counter or online pills. There's a huge market of almost 200 million. And when we found them, they're all unusual names like RU21 and Chaser, Uzis, very interesting names. And when we looked at the composition, they all seem to have very strange compositions like activated carbon and dextrosuccinic acid and zeolites of all things. So, and common to all these are that you take one pill before the session, one during, and one after the session. One pill before the session is okay, but now during and after, I don't think it's a very great idea. It will not work. So we said, okay, let's go back. Let's see what exactly happens in the target. So alcohol gets converted to acetaldehyde, alcohol dehydrogenase. It is NADPH reaction, NAD reaction, and aldehyde gets converted to acetate. Aldehyde dehydrogenase is also an NAD reaction. So basically,
In today's world, your workday doesn't always happen from 9 to 5. And it doesn't just happen within the confines of a cube or an office.
So now it's visible, sir.
Okay. Yeah, there. Just sorry about that. You're able to see the slides now?
Yes, sir. Yes, sir.
Okay, fine. Yeah, sorry about that. Yeah, so these are the kind of products that we have now prepared. The technology for this will be available very soon. It's a hango coated chili peanuts and chili cashews. Yeah, we've sort of found out that about 10 pieces of 15 pieces of chili peanuts and 10 pieces of this thing should be able to deliver about 500 milligrams, and about 5 grams of this SOMF should be able to give you the required amount at the end of the session. We also did. There's one person who was interested in trying it in Vietnam. Vietnam is a Southeast Asian country, and they have a problem where people experience a red face when they drink alcohol, and that's because of what is called as acetaldehyde poisoning. So we checked whether we sent it to them, and this is the report that they sent. Um, and yeah, and they said that the amount of redness actually goes down, and we found it quite encouraging because that's one of the big problems that they have in that country. And the amount of and we also did not have any hangover, even though they had about 10 to 12 bottles of beer. Their beer is around 350 ml with 5% alcohol. So each participant perhaps consumed almost 4 liters of beer, which is equal to 6 big bottles in India. So, which is actually quite encouraging for us that it actually works even in binge drinking. And yeah, and then, but this is not, those are not good enough for the results. So we had to do a clinical trial as well by a third party. The person who has taken the license is in Bangalore. We have sold us to a license. We already licensed this particular process. And the licensee is in Bangalore. We are in Mysore. The trial was done company. So that's actually a third party valuation. The results are actually placed on the CTRI website. So once you've done this, then how does your product actually stand in the market? So we did this competitor analysis, and then we found that you have some of these are the price of the Indian products. This is capsule is by Party Smart. This is a Himalaya truck company, and this is the only one that is taken before the capsule taken before the session. All these are after the consumption of after the session is over. And of course, this is that most popular one, RU21, is about $1 each. It has ingredient has activated charcoal, and you have to take one before, during, and after. So essentially, there are no snack-based products that can be consumed anytime during the session. So ours is sort of unique. How big is the market? The potential. So if you were to look at these two are the WHO figures. So 30% of the alcohol is consumed in India, of which 37% are moderate drinkers. Out of these 37%, 90%, assuming that they will have snacks with a drink. Typically, that is an Indian way of doing things. And out of this 90%, 60% will be interested in this product. And at MRP at this is the market size. It's huge. So this place for everybody. So now this is the name of the company that to whom we have a license. It's called Praras Biosciences. It's a biotechnology company. It's in Bangalore. It's a business to business ingredient. So they just produce the A-Hango and then they can be given to pharma sector, snack sector, beverage and this thing. And these are some of the retail units where they can be sold. It can be sold online on liquor store. This is the fine dining restaurants. This is the home party or ready-made snacks. And this is pub food. So it can be sold anywhere. This is the B2C. So, and since this is and as I said, the market is big and there are a lot of manufacturers for B2C. So we are now waiting for for this particular license to be given. So then it becomes easy. I think to be able to see this in the market quite soon. And yeah, so this is how we have taken the idea from an idea to a market. This is how we have taken translational research. So this part of the talk is over where again we would urge anybody to drink alcohol, to please don't drink and drive and drink responsibly. Okay, the next one is a very interesting product called Dolly-Mix. And this CFTR was established in 1951. So one of the first problems that we started was on idli, and this paper in 1960, studies on idli fermentation in a CSR journal. This laid the foundation for the dry mix composition, which you find all these products in the market now is actually the root paper for this is this one. So much outside. So we had our own bulletin and we would publish all the results there. In this particular journal, we published a paper which is the foundation for a ready-to-eat use dosa batter. The one that you find nowadays. This was released in 2001. More than 50 entrepreneurs took it. Now we've converted into free technology. You just have to register on the CFTRI website and you can download the project document. So these are some of the products that are available in the market, which are based on CFTRI technology. So once having been in this particular thing, we were very surprised when we learned this. Just the Bangalore city under the release per day, and just the highway of 138 kilometers between Bangalore and Mysore is about 1.2 for the weekend. CFTRI in its last 70 years of its existence, we have almost 17,000 research articles in our library. So we just sort of sat down and then we just sort of went on to see who all has worked on what. And this particular PhD thesis in biochemistry of CFTRI in 1978. This was very interesting and we utilized this. If you notice that this is steaming temperature, you get almost a 15% swelling and the solubility is almost quite high. So we said, why don't we use this particular grain as dolly mix and see whether we can actually enhance the number of idlis. So with that in mind, we designed this particular product called dolly mix. Dolly mix is dosa idli mix, and it's designed to give soft idlis, increase the number of idlis by 20%. Essentially, it is this part that helping it to increase. And you can actually add this in any proportion of rice and urad dal. Rice and urad dal, probably this thing is around only 3 is to 1, 2 is to 1, 4 is to 1. It depends on it's actually it is driven by economics rather than any other thing. Because urad dal is more expensive than rice. We found that it actually is good and it works. Draws in a lot of water actually. So this we found that the volume actually also increases upon fermentation. The texture of the idlis are also much better than the one that you get than traditional idlis. We tested this in commercial establishments, including CF Terra Canteen. We had 6 households and also put it in dry mix. This is the formulation that they did, and he almost got, he could have got more. Actually, he didn't add more water. He sort of didn't understand it quite well. So he would have got another 20 idlis more. He would have made more money. We then gave a feedback. We asked all the people in the field, the city to give their feedback. In fact, our Dr. Parmesh, who's there with you, he was one of our guinea pigs. He was also there, and I don't know what he gave it, but I think yeah, he was also there. And we tested this. It should not be that this works only in Mysore. They were tested in Hyderabad, in Pune, and Bangalore. We have licensed the technology to the Taj Group of Hotels in Hyderabad. So next time you're in Hyderabad or go to, say, after the pandemic, perhaps they should be able to start using our product in their market. This is how it looks. This is the control idli, and this is the Dolomix idli. It's quite, and we suggest that this is not exclusive. This is non-exclusive. So anybody who's interested, it's 40,000 rupees. You can actually put this in bulk to bulk pack, B2B packs of 25 kgs and sell it to street vendors, and they make a lot of money. They'll make more money. This part is the Dolomix idli part. So this is interesting. This is one of those ideas that came when you're sitting down and browsing on the internet mindlessly, and you keep going somewhere, and so we got this particular idea. We were actually looking at antioxidants in cancer treatment. The search pattern went everywhere, and finally, this sort of attracted our attention. That tumor GSH depletion under in vivo conditions has potential benefits for cancer therapy. So we went around looking for the GSSG, GSH glutathione part. And all these actually articles, they all talk about particular this one, where the GSH levels goes down and increases the oxidized glutathione and push it towards apoptosis. I don't work on cancer, and don't. But then when I saw this, I said, okay, let's see if we can do something on this. And when we started and discussed, we have one or two colleagues who work on. So we discussed. This is fine. One of the actual current approach for using this would be that you deplete the GSH availability through its biosynthesis, and therefore reduce the ratio of GSH and skew the ratio. This is also a very important molecule which is needed. So we said, why don't we convert whatever GSH is there and convert it to GSHG and push it directly inside and deliberately change the whole ratio? Can we do that? So said, okay, but then it's easier said than done because they are extremely, they have a guerrilla warfare technique. They are very wily. They have a lot of escape mechanisms. So you can't be pussyfooting around them. And so you need to change the rules of engagement. You have to treat it as an enemy. So we then studied all the war military strategies that were there, including from Mahabharata. The one that we actually found was very interesting and apt was this Pulhan military formation used in the Battle of Visalvana. And the first Anglo-Zulu war. the idea was to encircle the enemy, pin them down, and kill them. So we found this very interesting, and we said, okay, let's use this. This gentleman is Chief Shaka Zulu. He was a chieftain of a very small tribe, and he developed this particular method called Impondo Zincomo, the Bullhorn Formation, and created a huge Zulu empire. Basically, what he did was he divided his army, and he had young fellows running and encircling the enemy. And you had one group of soldiers then getting inside and pinning the enemy down, and you have the more experienced ones killing it. And this helped him to develop the whole empire to a Zulu kingdom, from a small tribe to a kingdom. And when it became big, it attracted the attention of the Britishers, and they had this first Zulu war where they used the same technique. And these people handed the British Empire one of the worst defeats in history in the British Empire. So, 100 years later, the Hollywood made a movie out of this with one of the greatest actors in those times, Sir Peter O'Toole and Bob Hoskins. So we then had, and we said, okay, let's look at this part, and we made our own formulation. And we created a formulation which the mode of action is something similar to this. This cartoon is actually made by two of our project assistants, extremely good artists. So we did this, and we taken a copyright on this. Basically, what happens is you suddenly skew the ratio of the CSH and CSSG ratio between ships, and once this comes in, the enzyme starts getting activated, and we ensure that it is sort of inhibited, both this by NADPH, and we inhibit this with our formulation ingredients. And once this is done, it starts wearing. So the problem remains, but there is no solution to this as far as the cancer is concerned. So it starts veering towards apoptosis. At this point in time, these guys come in catamarans, and they finish off the job by putting in and pushing it towards, shoving it towards apoptosis. And notice these guys in blue. These are our allies. These are the guys who are the drug guys who take doxorubicin and walk towards DNA. So essentially, we were looking at two theaters of war. One was this, and the other one was towards DNA. And both of them ensure that there are two different theaters, those separate. We don't want to create the same thing. So we said, okay, does it actually work? Looks extremely good, but then does it actually work? Since it was an offensive strategy, again, we changed this particular way in which we looked at it. Since this was an offensive strategy, we needed to develop a strategy where the normal cells were saved to prevent collateral damage. And then, so essentially, we tested this on both normal and cancer cells. And the way we did it was through a new area called genetic algorithms. Genetic algorithms is generally used in telecommunication, and so we're using it for the first time for this. And once we have, so what we did was we ensured that the viability of the normal cell was greater than 60%. But at that particular concentration, the viability of the cancer cell should be less than 40%. So there should be a difference of 20%. With this in mind, we did about 3 rounds of formulation, and this is how it is the final ratio. And so you notice that this is the highest. You're getting very high viability of the normal cell, and then you're getting very low viability of the cancer cell. Once this was done, does it actually work on individual? Does it better than individual, or is it combined together? It's more effective. So yes, it is more effective when it is all combined. In the presence of a drug, what actually happens? So we use this in the same cell lines on cancer cells. You see this is 50, I see 50 value, we get this. And then if you combine all these in combination, cisplatin gives you almost less viability. So yeah, it sort of works. Then we put that on animal trials, and we put it on animal mouse. We used EAT bearing mice. This is the control. This is a tumor one. This is a tumor with the medium dose. This is the medium dose that we do. And this is the tumor with the drug. And this is a tumor with the drug and low dose of this. So you notice compare E with D, E with B, and E with A. So it almost works out. And 250 milligrams is the one that actually we said per kg body weight. It translates to human equivalent dose of around 15 grams. That's about 3 teaspoons, which is good enough. That's about 15 grams. Yeah, so this should be good enough. So, and this is the result. We gave the dose every alternate day after the sim was done. This is the body weight, or this is a tumor, and this is with the drug, and this is in combination with the drug. And you notice how the whole thing is. And this is the survival. This is a Kaplan-Meier plot, and this after implantation, 20 days, the cancer cell they died, and the drug they died after 32 days, and in combination with the low dose, it died after 36 days. So there is a good lifespan increase as well. So we have now, yeah, this is how. These are the only results. Basically, what we are trying to achieve now is we want to have a food supplementary, a universal delivery that can be compatible with current treatment regimes. There are many ways, there are many cancers. So we thought if we can actually come up with a food supplement that actually helps to kill all sorts of cancers, we do all. And the outcome would be that most of the foods that are there in cancer treatment are all meant for side effect, overcoming the side effects rather than. So we thought instead of doing that, we may as well have one that actually aids in the treatment. We have patented this, and we are awaiting expression of interest for any of the startups, big companies to come up and take this forward. So yeah, that was that part. The final part was, can we develop new global foods? How do we can we do some new global foods as another way of fortification? You've all in microbiology and all, you've all read about Camembert cheese and Roquefort cheese. Essentially, these are the cheese that are grown using these organisms. And this is in Europe. And we have Indian cereals and grains like millets and all. So we thought, can we actually create new foods where you grow these organisms on Indian this thing and create new foods where we can actually get all sorts of newer types of metabolites that can perhaps be very useful. So we did this and it sort of worked. Because in Europe, they are used to metabolites of this. In India, we're used to metabolites of this. Can we actually create newer and newer metabolites, molecules that can be useful? So we developed Roquefort on Kodo millet. Roquefort is the one that gives that blue thing. But Neelamadana, Neelam is to Sanskrit, and Madana Kuruth is Kodo millet. So it's Neelamadana and Kaval, similar. So we grew this, and it's quite safe actually, because these are grass organisms and it works out quite safe. Yeah, this is how it looks. And we found that the advanced glycation end products, diabetes complications, actually goes up quite high in emissions. So it's useful in diabetes complications. And we also did this is a very good PhD study if somebody is interested in taking this forward or any other thing. This is the HPLC for unfermented foods, and this is the fermented food. This is HPLC profile. Take the unfermented one and you pass it through the amylase phase, gastric phase, and intestinal phase. This is the in vitro digestion system, and look at for the change in the metabolites. You then convert all these metabolites into, look and see how the molecules are actually getting modified to give you the biological activity. Use these to link it to MSMS, and then perhaps you could be able to do a better analysis of the kind of molecules that it is being transformed. And why I said for fortification was perhaps making the calcium more available, potassium and sodium ratios also sort of gone up. So this is quite useful, and it increases overall flavonoids content. Yeah, so this is looks like safe, but then as of now, we have published this work in Journal of Functional Foods 2 years back or something. So it's a good area to work with actually. You can actually create more and more. Fungal fermented foods now is available in California, I think. FDA has given approval. to grow that as well as a medical food. Yeah, this is a team for all these products. These are all the people who have actually worked. I have not divided them into scientists and technical officers and students are all mixed up. I don't want to differentiate any of them. Each of them have contributed in their own way. Yeah, so we don't want to separate them by the designation. And of course, most important was all the tasters, especially the alcohol hangover feedback. Every Friday evening, afternoon, we would get a call, get a sample. So we would send them samples, and Monday morning, we would get the feedback. Based on that, we would go ahead with any modification that would be required. We would take criticism very seriously, not personally. Because at the end of the day, what we are interested in the product. So when I actually called the people for a meeting to see if they could be interested, nobody turned up. Then only 2 turned up, and finally about 100 people asked for samples later. So still have the taboo about drinking. So, yeah, so this is I come to the end of my talk. So thank you.
Thank you very much, sir. Sir, it was really a wonderful session, and sir, we had few questions from participants. Sir, can you hear me, sir?
Yeah, I can hear you. Just kindly post.
Hello.
Dolomix contents, yeah, okay. One question is Dolomix contents, yeah, these are based on processed grains. I will not be able to give you much more. How much is the cost expenditure per 100 grams of? It's somewhere around, including processing and everything, I think works out around 50 or 60 rupees a kilo or something. But I'm not, because these are cyclical figures, because the grains actually that you get to process them, they are sort of cyclical thing. Perhaps if you can write to TTB, they'll be able to tell you how much we get.
No, you might go off nowadays.
You're able to hear me?
Sir.
Okay.
Sir,
that
there
is
is one more question. Sir, one more question is there. Sir, what made you invent Ango product? Is there any clinical studies, especially in human?
Yeah, it was there. We did a clinical study. I told you the CRO study that was done in Bombay. It has been done because without that study, nobody will give you a license. So that has been done. Yeah, it has been done by a third party. This O3, one more is there. O3 cancer cell lines, as to which organ. O3 is actually we purchased it from NCCS, or I think it's purchased from the person purchased from NCCS, and these are human ovarian cell lines.
Sir,
sorry. Yeah, please.
Sir, another question is there. During this COVID pandemic situation, being a food scientist, what is your suggestion for diet where it can boost our immune system to fight against COVID-19, sir?
You will have to work on basically. Again, I think I mentioned that in the starting. You have to look for you have to look for one good way to start would be sports recovery drinks. These are the good ones that actually help you get your energy back. Immunity boosting and all, yeah, you need to. We have one or two formulations that have done, not a spirulina, but non-spirulina formulation.